This type of cross, where the F1 is two hybrids, has a special name. It's called a monohybrid cross. Monohybrid crosses will always have a standard ratio, unless something strange is happening. So if you see a monohybrid cross, you can expect a 3-to-1 ratio of the dominant trait to the recessive trait. If that doesn't happen, say you get a 2-to-1 cross, this suggests something else is also going on. There are other types of special crosses too. For example, test crosses work a specific way too. In a test cross, you set up a mating between an individual of unknown genotype and a homozygous recessive individual whose genotype is known. This time, instead of peas, we're going to think about it in dogs. The color of a lab's coat is determined in a dominant recessive fashion, where black is dominant to brown. In this situation, we don't know who the parents of the black dog are, and we don't know if the black dog is homozygous for black or not. If we want to answer this question, we can cross the black dog to a recessive individual. Depending on what the black dog's genotype is, we can expect different outcomes. For example, if the black dog is homozygous dominant, then it will give the dominant allele to all of its children. This will result in all of the puppies from this cross being black. But if the dog is heterozygous, half of the puppies will get the recessive allele and be brown. So a test cross is a specific type of cross used to determine the genotype of an unknown individual. It always involves crossing that individual to a homozygous recessive known individual. If the recessive trait appears, then this one was heterozygous. In this type of cross, we would always expect to get a 1-to-1 ratio if the heterozygous individual is present. So these are Mendel's standard ideas about dominance and recessives, about where alleles come from, what an allele is, and how to understand what a cross would be for a single gene. And most of this is a review. But there's more going on than you might think. Mendel's laws are valid for a lot of things, but they are extremely simple. For most sexually reproducing species, these laws are true. But genotype often does not dictate phenotype in such a simple way. We also know that Mendel cooked his books. His ratios are too perfect. To make it more easy for him to explain what was happening, he also avoided any traits that didn't behave exactly the way he thought they should. For example, let's say we were to do this cross. When it comes to plant colors like white and red, most of the time when we cross them, we would expect, given Mendel's laws, to get flowers that are all red. They would be heterozygous, right? But that's not usually what happens. Instead, the heterozygous individuals are a new color. When new colors or new phenotypes arise, this is caused usually by something called incomplete dominance. Incomplete dominance happens when one copy of an allele is not enough to give the original phenotype. In this situation, we can see it come back when we do the cross with the F1s. We still get 4 outcomes, but this time all of the heterozygous individuals look different. Incomplete dominance will give us a phenotypic ratio that looks just like the genotypic ratio of 1 red, or homozygous dominant, to 2 pink, or heterozygous, to 1 white, or homozygous recessive. The first tip-off that incomplete dominance is present is the appearance of the new phenotype. Another difference is a process called codominance. Codominance happens when neither allele is dominant over the other. The classic example of codominance is blood types. The ABO blood group in humans is controlled by 3 alleles that produce a total of 4 phenotypes. This is because 2 of the alleles are codominant. The alleles are IA, IB, and lowercase I. Both of the A and B alleles are dominant to the lowercase I allele, but these two are codominant to each other, creating heterozygous individuals that display an AB phenotype, where both are equally expressed. This is because these genes and their alleles control which types of identifying markers are on the outside of blood cells. For example, individuals that contain at least one copy of the dominant A allele will produce an A carbohydrate, identifying this as belonging to an individual with type A. Individuals that have at least one IB allele will produce the B carbohydrate, and individuals that have IA and IB will produce both, whereas individuals with the recessive allele produce none. This is why you cannot give blood to certain people unless it matches. Each of the carbohydrates is used by the body to recognize self. If the blood given to an individual produces a carbohydrate that the body itself does not, then it will be recognized as a foreign cell and destroyed. This is why we think of O as the universal donor. Because no carbohydrates are present on the outside of the O blood type, the body will not recognize it as foreign. AB is considered the universal recipient for the same reason. Because this individual recognizes both A and B as being self, cells that have only A are accepted, only B are accepted, both are accepted, and neither would be accepted. There are a few other things that determine whether or not blood type will match, but this is the primary one. So let's think about how we could answer a question using codominance. In this question, Maria has type O blood and her sister has type AB. The girls know that both of their maternal grandparents are type A. But what are the genotypes of the girls' parents? To approach answering a question like this, it's a good idea to diagram what you know. For example, we're aware that Maria has type O, that her sister has type AB, and that we know the maternal grandparents are type A. Here's Maria and her sister, their parents, and their maternal grandparents. We know that Maria is type O. That means her genotype has to be 2 recessive eyes. We also know that her sister is type AB. To be type AB, she must have one copy of each of the A and B alleles. This tells us that the parents have to be able to give these combinations. Looking at this question, we see that for Maria to have this recessive blood type, both of her parents must have the recessive I allele as one of their alleles. In order for the parents to have a child that are AB, one must have the A allele and one must have the B. But how do we know which one? What other information were we given that tells us which parent is type A and which parent is type B?
grandparents?
That's right. We were told that both of the maternal grandparents have type A. This means it's very unlikely that the mother has type B. So she is probably the individual carrying the dominant A allele, which means that the father is the one carrying the dominant B. We can also check if we were to make a Punnett square about this, we could put the mom on one side and the dad on the other. If these are the mom's alleles and these are the dad's alleles, then the possible outcomes are like this. Do we see Maria and her sister in here? Sure. Here's Maria, and here's her sister.